Hello, this topic focuses on the structure of cells and functions of organelles located within cells. The presentation is intended for students taking Anatomy and Physiology 1 with Gina Piscatelli at Madison Area Technical College. Please consult your syllabus for the reading assignment and study question that goes with this topic. So to begin, let's discuss the intricate internal structure that cells have. This structure enables them to perform various functions in the body. The figure shown, for example, on this slide shows some of the internal structure in a cell that lines the pulmonary artery. The green labels actin filaments, which is part of the cytoskeleton. These are proteins that help give the cell structure and form. The blue labels the nucleus, which is where DNA, the hereditary material, is located. And the red labels many mitochondria, where cellular energy is produced. Cells do vary in shape, size, and internal structure, depending on the specific cell and the function it performs in the body. The diagrams of specific cell types that are shown on this slide reflect some of these differences. In the upper row from left to right, we have neurons on the left with elongated extensions that can conduct electrical signals. The picture in the middle contains epithelial cells, which of course do not have these elongations, but they are tightly adhered to one another to protect and line an organ. The third picture on the upper right shows red blood cells that carry oxygen. The lower row from left to right depicts first an elongated muscle cell that can shorten during contraction, and then in the middle is a row of thin or epithelial cells that form a thin sheet. These cells allow oxygen and carbon dioxide to cross a barrier. The third picture, the lower right, shows white blood cells that secrete substances that help provide immunity and protection to us. Regardless of cell type, though, most cells have some basic structure in common, including a nucleus that houses DNA, cytoplasm that consists of cytosol, a viscous fluid containing small internal structures called organelles and cytoskeletal proteins, and then finally a cell membrane. This forms the outer boundary of the cell, and it most importantly interacts with its environment in which the cell exists. Please note, however, that mature red blood cells do not contain a nucleus. The nucleus and DNA is lost as the cell matures. But we'll look at the nucleus first in more detail. Most cells have one nucleus. Other cells have more than one nucleus. They're said to be multinucleated. The structure of a nucleus includes the nuclear envelope, which controls the passage of material into and out of the nucleus to protect DNA. Some substances can pass through nuclear pores, which are shown on this diagram. Within the nucleus is the nucleolus, a dark staining region, shown here in blue, that stains this way because it has RNA and protein in this location. This is where ribosomal RNA is made before it exits the nucleus to become part of a ribosome, which is located in the cytoplasm. And finally, within the nucleus are thin dark lines of chromatin. This is a form of loosely coiled DNA that is located within the nucleus and surrounds the nucleolus. This slide shows a picture of the nuclear membrane in more detail. Note that there are two phospholipid bilayers, shown in blue, and in this instance we see the structure of a nuclear pore through which RNA can move. In contrast to this nuclear membrane, the cell's membrane has only one phospholipid bilayer. I'd like to expand on the structure of DNA and chromatin that I mentioned previously. This is the genetic material found in human cells and consists of 46 strands of DNA wound around histone proteins. These 46 strands are referred to as chromosomes. But in a non-dividing cell, the 46 chromosomes are not visible under the microscope because the DNA is loosely coiled and in the form of chromatin. So in this case we refer to the genetic material as chromatin, and it's shown in your diagram on the slide. But right before a cell divides, and then throughout cell division, this DNA supercoils and becomes tightly wound around the histones. This prevents the chromosomes from fragmenting as the chromosomes are moved around to be put into a new cell. This slide shows a karyotype. In this case, chromosomes are displayed in a linear format, and it enables geneticists to look for damage and any abnormalities. Because the chromosomes are visible, we know they're tightly coiled or supercoiled and can be seen under the microscope. Notice that they're arranged according to 23 pairs. Each pair of chromosomes codes for the same kind of information. Beyond the nucleus is the cytoplasm, which consists of all the material located between the nucleus and the cell membrane. Cytoplasm consists of both cytosol, fluid, and organelles. Cytosol is viscous, but it is made up of 70 to 80% water with many solutes that make it viscous. These solutes include salts, proteins, lipids, and carbohydrates, to name a few. The organelles are suspended within the cytosol, and each organelle is specialized to perform a specific function in the cell. Many organelles are membranous compartments that contain molecules for certain chemical reactions. Other organelles are aggregates of organic material without a membrane. In addition to being the fluid in which organelles are suspended, the cytosol has several functions. These functions are related to the enzymes that it contains. Some functions that cytosolic enzymes perform include breaking down of compounds or producing ATP by the process of glycolysis. Cellular organelles located within the cytoplasm, as I mentioned previously, are of two types: those that are membrane-bound, or membranous, and those that are just aggregates of molecules without a phospholipid membrane. Those that have a membrane include the endoplasmic reticulum, which is subdivided into a smooth region and a rough region, the Golgi apparatus, also called the Golgi body or Golgi complex, mitochondria, lysosomes, and peroxisomes. The organelles that do not have a membrane but are aggregates of molecular structures are ribosomes, centrioles, and the cytoskeleton. I will begin discussing the function of each of these and start with the membrane-bound organelle that is closest to the nucleus. That's the endoplasmic reticulum, shown here in blue. The endoplasmic reticulum is a membranous system of sacs and tubules. Its cavities are called cisterns or cisterna. It has two distinct divisions, the rough ER and the smooth ER. The rough ER is noticeable because it has ribosomes on its surface. The smooth ER does not have ribosomes on its surface. Now the rough ER and the smooth ER have separate functions. The rough ER is associated with protein trafficking and protein synthesis because the attached ribosomes will make proteins and insert these proteins into the ER cisterns. Ultimately, the proteins that enter the rough ER will be exported from the cell. Within the rough ER cisterns are enzymes, which can glycosylate proteins. This means that a carbohydrate will be added. The proteins that are made inside the rough ER, or made by the ribosomes and inserted into the rough ER, are packaged into transport vesicles, which will be budding off of the ER and travel in the cytoplasm to the Golgi apparatus. As I mentioned, these proteins are destined to be secreted from the cell or may be attached to the cell's outer surface. In contrast, the smooth ER lacks ribosomes on its surface. Therefore, proteins are not made here. Instead, the smooth ER contains enzymes that synthesize lipids, including steroids like cholesterol. The lipids made in the smooth ER will also be put into transport vesicles that will bud off of the smooth ER. In addition, the smooth ER is also a site of calcium storage and detoxification of certain drugs, pesticides, and cancer-causing chemicals. This slide shows the process by which a transport vesicle forms by budding off of the rough ER in this case. A similar process happens with vesicles that bud off of the smooth ER. In both cases, proteins or lipids within the ER are contained within transport vesicles when the ER membrane invaginates. The vesicle then constricts to the point of separating from the main ER organelle. Any proteins or lipids inside the vesicle at this time will then be suspended within the transport vesicle. Depending on whether the vesicle formed from the rough ER or the smooth ER, and what type of molecule then is contained within the transport vesicle, that vesicle may fuse with the Golgi apparatus. It could also fuse with the cell's membrane or a lysosome. So the Golgi complex or Golgi body is made up of hollow flattened sacs. The membranous sac closest to the endoplasmic reticulum is called the cis-face. The membranous sac farthest from the endoplasmic reticulum, but closest to the outer cell membrane, is called the transface. Proteins and lipids move through these sacs to be sorted and modified. For this region, the Golgi complex is sometimes called the post office of the cell. So one of the major functions of the Golgi complex then deals with the movement or trafficking of lipids and proteins, making sure they get to the proper location either inside the cell or outside of the cell. The way that this trafficking occurs is that first a transport vesicle that budded from the ER merges with the cis face of the Golgi. This is shown in your picture as the blue transport vesicle fuses with the cis face, dumping its contents into the lumen of the first Golgi sac. These lipids and proteins are then exposed to enzymes within the Golgi, which can add carbohydrate or glycosylate the molecules. The glycosylated lipids and proteins will move into the next Golgi sac until they reach the trans phase. As the lipids and proteins move through the sacs, they are sorted and grouped according to where they will ultimately be sent. This is why the Golgi complex is called the post office of the cell. Finally, at the trans phase, secretory vesicles containing the modified proteins and lipids bud off of the Golgi apparatus. The secretory vesicles can move within the cytoplasm along cytoskeletal proteins of the cell. Most often, the secretory vesicles will then fuse with the cell membrane. The contents then, the proteins and lipids, will either be released into the interstitial fluid surrounding the cell or be attached to the cell's outer cell membrane. We will review this process again after we look at the structure and function of ribosomes. Ribosomes are an example of non-membrane bound organelles. There are two types of ribosomes based on their location within a cell. One type is called free and is suspended within the cytosol. The other type is called bound and is attached to the rough ER. But both types have the same structure, composed of two subunits, shown here as pink and dark purple. Both subunits contain two types of organic compounds, ribosomal RNA and protein. There are similarities and differences between the function of the free and bound ribosomes. Both free and bound ribosomes perform protein synthesis. That is, a ribosome is able to assemble amino acids to make a polypeptide. It does so by reading a copy of a gene, shown here in green. This copy was made in the nucleus and codes for a particular protein. But the type of proteins made by free ribosomes differs from the type of protein made by a bound ribosome. The free ribosomes remain in the cytosol and synthesize proteins that will also remain in the cytosol. The bound ribosomes, on the other hand, synthesize proteins that will eventually reach the cell membrane. This occurs by ribosome docking onto the ER. And as it synthesizes the protein, the ribosome inserts the protein into the lumen of the ER. Eventually, a transport vesicle containing this synthesized protein will bud off of the ER and travel to the Golgi, as I have discussed previously. The picture here shows the process by which a ribosome synthesizes a protein that needs to be inserted into the ER in order to get into a secretory vesicle. Protein synthesis always begins with a free ribosome. If this free ribosome detects a signal sequence, which is shown as pink in this diagram, it will dock onto the ER. Notice that the copy of the gene is green, the ribosome is purple, and it is freely suspended in the cytosol. It's currently synthesizing a polypeptide with 3 amino acids shown as the signal sequence. When this signal sequence is present, which is not true of all proteins, the ribosome docks onto the ER, shown to the right in the picture. While it's docked on the ER, the ribosome continues to make the polypeptide, and as the polypeptide gets longer, it will be inserted into the lumen of the ER. Therefore, to summarize the function of lipid and protein transport by both the ER and the Golgi body, let's look at this diagram. Lipids are made in the smooth ER. On the picture it says lipids are made here. That's the smooth endoplasmic reticulum. Proteins are made by ribosomes and inserted into the rough ER. On the diagram is a label that says proteins here. Both lipids and proteins leave the endoplasmic reticulum in transport vesicles, labeled transport vesicle on the diagram, that have budded off of the ER. This transport vesicle then travels to the cis face of the Golgi body. It fuses with the Golgi's membrane at this cis face, thereby releasing its contents into the first sac of the Golgi. As the proteins and lipids travel through the adjacent sacs of the Golgi body, they are modified by enzymes, which frequently glycosylate them, and they are sorted. Once the modified lipids and proteins get to the transface of the Golgi body, they are trapped into secretory vesicles which bud off of the transface. A secretory vesicle then travels to the cell membrane and releases its contents. The contents of that secretory vesicle, destined for the outside of the cell, may be released freely into the extracellular fluid, but sometimes the molecules stay attached to the cell membrane. We have covered a lot of cellular structure and function, including the nucleus, the nucleolus, DNA, both the rough and smooth ER, the Golgi body, and the ribosomes. This may be a good opportunity for you to look at your worksheet or assignment and answer questions pertaining to these organelles. When you are ready, you can proceed with the rest of the presentation, which discusses the membrane-bound mitochondria, lysosomes, and peroxisomes, as well as centrioles, the cytoskeletal proteins, and finally the cell membrane. So let's continue on with discussing the structure of cells, starting with mitochondria. Most types of cells have many mitochondria. These organelles contain enzymes that contribute to the production of ATP. Therefore, we often say that the function of mitochondria is ATP synthesis. The structure of the mitochondrion is unique. It has two phospholipid bilayer membranes, one called the outer membrane, the other called the inner membrane. The space between the two membranes is called the intermembrane space. The interiormost space, encapsulated by the inner membrane, is called the matrix. Notice that the intermembrane space contains ribosomes. This enables mitochondria to synthesize proteins, which will be needed as enzymes for ATP synthesis. These enzymes or proteins are coded by DNA that's located within the mitochondria in a single circular chromosome located in the matrix. The genes on this single mitochondrial chromosome code for proteins required for ATP synthesis. However, additional proteins and enzymes are needed for the mitochondria to fully function, and therefore need some of the genes in nuclear DNA. So both nuclear DNA and mitochondrial DNA are needed to code for the complete array of enzymes needed for the mitochondria to function. Notice the picture on the slide which shows the infolding of the inner membrane. These deep invaginations are called cristae and are an important site for the final step of ATP synthesis. Lysosomes and peroxisomes are both small membrane-enclosed vesicles that contain enzymes involved in the catabolism or breakdown of organic compounds. They do function a little bit differently. The lysosomes usually function in breaking down material that has been ingested by the cell. They also contain enzymes that can initiate programmed cell death, known as apoptosis, or the lysis of vesicles and old organelles. Peroxisomes usually function in breaking down fatty acids, and this process of fatty acid catabolism is known as beta oxidation. We are done with talking about membrane organelles, and now I will talk about those that are not membrane bound. Starting with centrioles, these are paired structures composed of microtubules made up of the protein tubulin. These microtubules are arranged in triplets so that each centriole has 9 triplets. The centriole and its surrounding area is called the centrosome, and this is the site where many more microtubules can be made. The extra microtubules that are made extend throughout the cytosol of the cell and become part of the cell's cytoskeleton. I will discuss them again when we discuss the cytoskeleton. As a microtubule synthesizing center, the centrosome, circled in fluorescent green on this picture, produces microtubules that will help distribute chromosomes during cell division. This is a picture of a cell that's undergoing cell division, with the chromosomes centrally located and attached to microtubules. The microtubules will draw the chromosomes to either end of the cell, in fact towards the centrioles, and be placed in an individual cell on each end. Other functions of microtubules that the centromere makes are found in cilia and in flagella of certain cell types. The picture on the left of this slide shows the arrangement of microtubules found in cilia and flagella. Unlike centrioles, these structures have paired microtubules rather than triplets. 9 pairs are found in the circumference, with 1 pair localized centrally. Both cilia and flagella are associated with motor proteins that cause the cilia and flagella microtubules to move. Cilia are found on some cells in the respiratory tract and oviduct. The respiratory tract cells are shown in the picture on the right. And when the cilia rhythmically move back and forth, they produce a flow of fluid that propels substances like mucus across the cell surface. Flagella, shown in the middle picture, are found only on sperm cells in the human body. When flagella move, they actually rotate, which provides a force that propels the sperm forward along the female reproductive tract. In addition to microtubules, the cytoskeleton has other protein-based structures called intermediate filaments and actin filaments. Because microtubules have the largest diameter, they're called microtubules. Actin filaments have the smallest diameter, and sometimes they're called microfilaments, filament being smaller than a tube. Intermediate filaments, as you may guess, get their name from the fact that they are intermediate in size. Notice that the intermediate filaments form a scaffolding within the cell, giving it support and structure. They're shown as orange in this diagram. Also notice that actin filaments are located just interior and adjacent to the cell membrane. These locations reflect the various functions. So each cytoskeletal protein type has a specific function within the cell. We've already discussed microtubules, which help with the movement of materials within a cell, like chromosomes. The top picture, though, in this case, shows a microtubule providing a surface along which a motor protein can move a vesicle inside the cell, such as a transport vesicle or a secretory vesicle. The middle picture shows intermediate filaments which provide the cell with strength and structural integrity. The bottom picture depicts actin filaments which can shorten and lengthen. When actin filaments lengthen or elongate, they will extend and the adherent membrane will extend with them. This allows a whole cell to move by a crawling mechanism, sometimes called amoeboid movement. This occurs particularly when white blood cells move from our bloodstream to a region of tissue that is inflamed. The top picture shows a cell that is not moving, but the middle picture shows actin filament elongation that extends the cell forward. The rear of the cell is pulled towards the leading edge by actin filaments rearranging. The last cellular structure to examine is the cell membrane, which has many functions. Some of its functions are obvious, such as defining the boundary of a cell. However, there are other functions which are not as obvious. Another is that it controls the passage of materials into or out of a cell, which is extremely important for water balance and solute balance. The membrane may allow some substances to cross and bar other substances. This is referred to as membrane permeability, and we will discuss it in more detail. Another role of the cell membrane is related to how cells communicate with one another. The cell membrane may allow direct contact with another cell, or it may receive a signal from a cell, bind to it, and this will trigger a response. This type of communication is called cell signaling. And the fourth function has to do with anchoring the cell in an extracellular matrix of proteins. First, let's examine the structure of the cell membrane, because membrane permeability depends on the structure of the cell membrane and is essential to the function of the whole cell. The cell membrane is largely made up of lipids. In fact, 98% of the compounds in a membrane are lipids, and only 2% are proteins that are inserted into this lipid membrane. There are also different types of proteins. Many membrane proteins span the entire width of the membrane and therefore are called transmembrane proteins, shown in the diagram as purple structures embedded in the phospholipid bilayer. The structure of a cell membrane is often described as a fluid mosaic. What this means is that it is a mosaic in terms of having multiple types of compounds present, and fluid in the sense that molecules or compounds can move or change position within the fluidity of the membrane. The purple transmembrane proteins that you see in this diagram can move about within the phospholipid bilayer. The phospholipids can also move. Another thing that you notice within the phospholipid bilayer is cholesterol, shown here in blue. Cholesterol influences the rigidity or fluidity of that phospholipid matrix, and the structure of cholesterol can be influenced by temperature, pH, and the degree of hydrogen saturation. So the amount of cholesterol and how stiff the cholesterol is will affect the phospholipid bilayer. This dynamic feature of cell membranes was discovered during an experiment in which two cells were fused together, and the location of membrane proteins were observed in the newly formed cell. First, a mouse cell's proteins were labeled with a blue fluorescent dye, shown here in the picture, the circular cell. And a human cell, shown here as a rectangular cell, was dyed for its proteins with a red fluorescent dye. The two cells were easily fused into one cell by just adding a spark of electrical current. The membranes will fuse. This cell was then allowed to incubate in a carbohydrate-rich environment, and over time the researchers noted that the blue and red proteins relocated. They were distributed all over the fused cell's membrane. If the proteins had not been able to relocate or to move within the phospholipid bilayer, all of the blue proteins would have remained within one region, and all the red proteins would have stayed on the opposite end in another region of the newly formed cell. Our last topic concerning the cell membrane and cell structure in general deals with the functions of cell membrane proteins. These membrane proteins might allow cells to interact with other cells or the extracellular matrix. So number 1 is listed here as adhering to the extracellular matrix. Number 2 is listed as interacting with other cells to form tissues. Third, a membrane protein can interact with other cells to communicate. And number 4, some membrane proteins perform chemical reactions. Number 5, some membranes form channels or pores that allow specific compounds to cross the cell membrane to get into or out of the cell. And then number 6, some membrane proteins are markers for a cell's function, it's the type of cell it is, or a marker as your own cell, known as a self cell versus a foreign cell. So we'll go over these different functions briefly. This picture shows how membrane proteins are involved with adhering a cell to the extracellular matrix. Notice that the extracellular matrix has collagen, elastin, and proteoglycan molecules that bind to the cell's membrane proteins. This keeps the cell in place. To form tissues, cells are attached to each other, sometimes tightly and sometimes loosely. This picture shows two types of cell-cell adhesion structures or proteins. One is called a tight junction. As the name implies, membrane proteins on two adjacent cells, shown here as green, form a very tight link that prevents the passage of compounds from getting in between two cells. This is particularly important for cells lining the digestive tract, so that things like stomach acid will not leak in between cells. Another type of cell adhesion structure is called the desmosome. In this case, the space between the two adjacent cells is much wider, allowing some materials to pass in between the two cells, but the membrane proteins that provide cell-cell linkage, shown here as red and purple, are very, very strong. These desmosomes are important to skeletal muscle and cardiac muscle tissues, so that contraction does not cause shearing and breakage of a tissue. This slide shows how membrane proteins can interact directly with other cells for signaling purposes or communication purposes. In one case, two proteins are directly contacting each other, and that could signal one or the other cell to perform a certain function. In the picture on the right, a cell's protein, transmembrane protein, has received a chemical signal shown in green. Binding of that chemical signal will cause the cell to perform some function interiorly. As I mentioned also, some membrane proteins might catalyze a chemical reaction, that is, act as an enzyme. This could happen on the surface of the cell or within the cytoplasm. On the left, the starting material outside the cell is roughly a square. After binding to the membrane protein, the compound has been changed and the product is circular. This has happened outside the cell. The picture on the right shows the same process, but it is occurring inside the cell's cytoplasm. Membrane proteins are very important to cell permeability because not everything can cross the phospholipid bilayer, which we will discuss in class. So these proteins allow transport processes. Because they allow water-soluble compounds to cross the membrane, also known as polar compounds to cross the membrane, and phospholipids do not. They only allow lipid-soluble compounds to cross. So these proteins that allow transport usually form either a channel or a gap junction. A membrane channel, shown on the left, is a transmembrane protein in one cell that allows a very specific substance that's polar to move into or out of the cell. The picture on the right shows a gap junction. This is a structure that occurs when two channels of two separate cells line up perfectly so that material can pass through both from one cell to another. One location in your body where this is important, where gap junctions are important, is in the heart. Gap junctions allow the spread of ions from cell to cell, allowing the cells to contract nearly simultaneously. Lastly, transmembrane proteins can function as cell identity markers. Indeed, different types of proteins are found on different types of cells. In terms of medical research, this allows scientists to identify the type of a cell by looking for the presence or absence of a certain protein. For example, an embryonic neural cell can be differentiated from an embryonic muscle cell by staining for or labeling with specific proteins for each. Another example that you might be more familiar with concerns blood types. Blood that is positive, either A positive, B positive, AB positive, or an O positive, has red blood cells that have a certain protein in their membranes called Rh factor. So positive refers to the presence of Rh factor. In negative blood, type negative blood, this protein is absent. In addition, we each have, as individuals, a set of proteins called MHC that are found on all our cells. Our MHC proteins are unique from all those of other individuals. Closely matching these proteins is particularly important in organ, tissue, and cell transplantation procedures. This completes the presentation on cell structure and organelle functions. Thank you very much.